Well, hello everybody. Thank you for tuning into this episode of the Too Bad For You podcast. And boy, do I have a real treat for you this time. In this conversation episode, I was joined by Dr. Florence Kleberg. She is a computational neuroscience. And let me tell you, folks, this one is all science. In the past, we've done, a mix of science and then talked about some science communication stuff or science and society stuff. This was all science and some hard brain science. So get ready. Florence was a really great person to talk with. She was super enthusiastic, easy to talk to. I really, really enjoyed it. Like I said, she does computational neuroscience. So this is like really, mathematical models of the brain, or computer models rather, of the brain. Um, Her work focused a lot on synapses. So synapses are the little spaces that neurons, send signals, neurotransmitters through, which then, make the next one fire the electrical impulse or not. And so it was really looking at like mapping all of these connections in the brain, all of these synaptic connections in the brain. And we kind of, one of her main things was really about how these things change. These connections, these sort of, if you think about it like as a circuit, these neural circuits don't remain the same over time in the brain, which is kind of a bit counterintuitive when you think about the brain storing information. How does it store a memory in these circuit pathways if the circuit pathways are always changing? So like what is then we ask the question, what is a memory if it's not like a stable section of neurons that are firing and encoding that information? This led into talking about artificial intelligence, mapping the brain, mapping the synapses, mapping the neural networks. Obviously, there's some artificial intelligence stuff there. It also went into a lot about epilepsy. And so this was when we were discussing the way in which neurons regulate their activity. So the way in which these synapses sort of remain in a stable network where they're not all firing at once. What are some of the mechanisms behind that? Because when they do sort of, if you want to call it overactive, that's how you get epilepsy. So there was a lot in this conversation, but I hope you enjoy it. I really, really enjoyed talking to her. Like I said, super enthusiastic, really great to talk to, really nice person, super smart. She had a lot to say. We didn't even touch on half of the stuff that I'm sure we could have, so hopefully we can do it again. Um, I'll leave it there. Enjoy this conversation with Dr. Florence Kleberg. We'll have the freak motif bring us in, as per usual, and then it's on to my conversation with Florence. Enjoy.
Yeah, Brad, thank you very much for inviting me.
Yeah, no problem. Thank you so much for doing the show. Thanks for being here. I'm excited to be back at the Frankfurt Institute of Advanced Sciences.
Studies.
Right. I always get that one wrong. And actually, this is exciting because you're a neuroscientist by training, yes. And I find that when people that are interested in the sciences, it's usually space and the brain. This is what people are so interested in. So now we have someone who can talk to us about the brain. And I was reading some of the papers that you sent along to me to sort of get a bit of background on what it is you do. And I mean, my wife does neuroscience as well. Mm-hmm. And what I've learned about neuroscience is there's a lot of different aspects. Like she does behavioral neuroscience. There's like chemical things going on in the brain. There's structural, the different brain regions.
Yes.
From my understanding, what it is that you're doing is looking at the circuitry, so the electrical circuitry of the brain. So maybe you could start there and give us what is the electrical circuitry of the brain? Synapses are involved, yes? Neurons?
Yes, exactly. So what I'm interested in is computational neuroscience. So that's actually a field in neuroscience where people use either advanced data analyses or make models of the brain by using computer programs.
Right.
And the latter is something that I've been working on more recently. And in a lot of cases, if you do an experiment, you you set up the experiment, you record the data, and then you analyze this, and you make some conclusions about what you've seen. But there's always kind of a black box problem involved in the experiment, because you cannot control everything in the experiment. You can't control every parameter. But if you create a model on a computer of the brain, you can set all the parameters to whatever you want, and you know what they are. And this creates kind of a unique opportunity for finding out what does what in a brain, what does what in an electrical circuit of the brain. So that's one reason why making a model of the brain or parts of the brain is very interesting opportunity for understanding what's going on. Things that perhaps cannot be tested yet with experiments, or parts of the brain circuitry that you would like to see tested next, but you would want to look at some specific aspects, and in order to guide or to propose new experiments, you could do first some modeling to look at what would be feasible, what would be interesting to look at, and already maybe cross out some things that are not possible or not very interesting.
Right. So when you're making a model of the brain, I mean, we know it's an incredibly complex organ, right? Like all of these circuits and everything. Like, what are we talking? Like, we're talking like billions of neurons or something, correct?
Yeah.
So how do you start then building a model that you could be interested or confident is sort of reliable?
Yeah, that's a very good question, and it's not easy.
I don't doubt it.
It's you can't model yet the entire brain, both based on what we know about the brain. Like, our knowledge is still not complete. We don't know every single thing about the human brain yet. And the other part is the computational capacity you would need to actually implement this model, to run it for long enough, and so on. So there are limitations in what you can do. And so you have to take a piece of this brain, some brain region perhaps, some type of cells that you would like to look at, and simplify this very much, and then create the model. And in the ideal case, you should have a model, and before you run it, you should already know what you would want to see comes out of this model.
So have like an expectation that if that doesn't happen, then you can either know that your hypothesis is wrong or that the model's not functioning properly?
Yeah.
Okay, yeah.
So it would be hypothesis driven. You can also do exploratory, but I find myself that if you do a hypothesis driven research, it's easier to really see what's going on. You get really something concrete that comes out of the research. You can say, well, this is true, yes or no.
Right.
Well, if you do exploratory research in modeling, then you can run things forever and you don't really know what it means.
Right, because I guess this is the thing with any sort of modeling, whether it's of the brain or climate or what, anything that modeling is used as, you kind of have to have the input characteristics as close to reality as possible. So you kind of, like you're saying, you have to have an idea of what to expect or how to.
Yes.
Right. Okay, that makes a lot of sense. Because that was even in some of the papers that you had sent along to me in the book chapter.
Yes.
It sounded like that's exactly it. You guys sort of had this expectation, or I'm not sure, you can correct me, I'm usually wrong about most things. So that, like you could see the synapses firing or a way in which they were firing. And then it was a matter of, okay, what modeling conditions would produce that. So you could take a picture of the brain, say, or like a picture of the firing of the brain. Picture's the wrong word, obviously, but you didn't know what combinations of neurons or synapses were firing that produced that signal.
Exactly.
And so the model was then trying to reproduce that signal, but because you made the model, you knew what went in. Is that?
Yeah. That's more or less it. Yeah.
Okay.
So yeah, like trying to understand in the case of the book chapter, like how can you get these bidirectional connections between neurons? This is something that has been observed in the brain. It comes out of some experiments, and then the way these models are used is kind of, you can see it maybe from an engineering perspective. Okay, we have point A, we want to get to point B somehow. What if it's this kind of mechanism? And then you put that in a model, and then you do get the same thing. You're like, okay, it could be this. Of course, it could be something else completely that we
Right.
don't know about, that we couldn't even conceive right now.
Right.
That's the trick with these models. You can never say like 100% like, oh, this is exactly what is happening, unless you can really show that nothing else is possible.
Right.
So that's kind of the downside of these kind of modeling. But just to get back to the electrical circuit you,
Right,
you
yeah.
mentioned earlier that I wanted to explain. So the models I've been working on with my colleagues, they are models of the brain that look at the circuit level, so the network of neurons in the brain that are connected to each other by synapses.
Right.
And how these networks function, what can they do, what kind of activity do they have, and importantly, what is the plasticity happening in these networks? And
Plasticity being like that they can change the route that they're using or something like this.
yeah. So
okay.
plasticity is something like, you can change something in the
Right.
connections between the neurons or in the neurons themselves that would alter how they respond to input, for example.
Right, right.
So one very well-known type is synaptic plasticity. So you have synapses between the neurons. It's how they transmit signals, one of the ways they do. And these synapses can become stronger or weaker over time due to various factors. Could be due to the activity happening in the presynaptic neuron, could be due to the activity in the postsynaptic neuron, could happen over time due to homeostatic effects or limitations of resources inside the synapses that use the same resources from nearby synapses, and so on. So there are many different types of plasticity have been shown in experiments, and by using these circuit level models, we try to implement simplified versions of these plasticity rules and try to understand what they do in these networks and what are the limitations of these rules, or what is something that maybe doesn't work well, and maybe we need some extra research, some extra experiments that are done on these synapses, and how can these networks of neurons learn something, how can they retain something, and how can they create memory? These are
Right.
very big questions, actually, that are all related to trying to understand this electrical circuit.
Because, yeah, so like when you're learning an activity or when your brain is, I guess, encoding and like storing a memory, that's going to require that the synapses are changing or doing different
Exactly,
things.
yeah.
So I guess your brain is constantly, these synapses are constantly changing.
Yeah, I was about to get to that. So your brain right now is different from now.
Yeah, that's
It's-
so
yeah.
weird to think about that my brain is changing.
Yeah.
Like it just it's one of those sort of trippy, mind blowing things. But yeah, it's amazing.
Yeah, it's constantly changing, and but despite the fact that it's constantly changing, there was also some consistency over time. We can have a certain memory, for example, of yesterday I went to a restaurant, had a salad, and then the next day you still have this memory.
Right,
Like, oh yeah,
doesn't
2
go
days
away.
ago I went to have that salad. So how can these memories be persistent at the same time, while we also know that there are constant changes happening in this circuit due to new memories coming in, new memories being formed all the time? I mean, you forget some things, but you also learn a lot of new things. And that all of that combined with the fact that at the synapse level, for example, the neuron level, there are a lot of molecules that have very limited lifetimes and that are constantly turned over. I mean, you've studied biology, so you understand this, right?
Yeah.
They're constantly being replaced, and how can you retain some stable memory in a
Right.
neural network if you constantly have to replace all these molecules the right way in the right places? It's really not understood how this can really be maintained so well. And then, of course, there's the question of, like, really, what is the memory?
Yeah.
We don't even know what the memory is.
I was going to ask. I wasn't sure if that was going to sound like too silly or too. But
No,
it is.
no, not at
It's
all.
a deep question, right? Like, what is it? What is it in the brain? Is it just the network of circuits? I mean, that can't be the whole picture, because we know now that these are changing. So
Yeah, they are constantly changing. There are synapses that are more stable over time compared to other synapses. So there's like a subset of synapses. If you look at some like brain region, for example, bowel cortex. So that's a cortex in a mouse where the whiskers are whisker information comes in.
Okay.
And if you look at the distributions of synapses in there, you can see that there are some that are very stable over time, usually the bigger ones. And you see a lot of them that like appear and disappear over time. And so these synapses and these, they're not very stable, actually, if you look at longer time scales. One of the things that experimenters look at for these synapses is what they call a spine. And the spine is kind of like a protrusion on the membrane of a dendrite of a neuron. So that's like kind of like where the neuron receives the synapses from other neurons. And these spines, you can actually image them using very sophisticated microscopic methods.
Yeah.
And you can then image them over time, like days and days and days. You can see, oh, this spine, was it there? Is it gone now?
And so those are changing too.
Those are changing as well. And they're also changing in a size-dependent manner. So that some spines, just the bigger spines, for example, have larger fluctuations or smaller fluctuations, depending on where you are in the brain. So it's a very complex picture with a lot of, there seems to be some kind of interplay between consistency and transiency, which we don't understand. And we need to find out how this works, how this can be maintained over time, and where the memory is encoded. Because there are experiments that have shown that if you, for example, rodents learn some motor task over time, and you image these spines that I just mentioned, and you look at the spines, you see new spines appearing when the animal has learned the task. And you're like, oh, okay, this is probably some sign of the new memory being formed. You can use a laser to specifically target and remove those spines that have just appeared, and the animal has forgotten the motor task. That was published a couple of years ago.
You can erase them.
You can erase the memory.
Specifically erase that memory.
Yeah.
Wow.
Which is
that's kind of creepy.
Yeah, it's creepy. Yeah. But it's interesting because that is at least one kind of concrete way in which we can see, like, okay, the memory is there, perhaps.
And this structure is involved.
At least this structure is involved. That's what.
In some way.
Yeah, obviously other structures involved as well, but yeah. So that's really where we're at, right now, in understanding memory at the circuit level. I mean, there are many other approaches. There's like the psychological approach where you let people, for example, do memory tasks and look at the brain activity. So I'm not saying at all that this is the complete picture, but yeah. We're trying to understand this at the circuit level.
Right, that's a goal. So I mean, again, from like the circuit level seems like, understanding that there's a lot of different things going on, but just the amount, like to take a snapshot of the electrical activity that is happening in a brain at any moment.
Yes. And
then trying to extrapolate some kind of sense out of that. You mentioned the computing power that's necessary. Like this must be, computing power increases all the time. So it's probably a rapidly advancing field. What you couldn't do a year ago, you can start to look at now. Yeah.
At some point, we might be able to really simulate, nearly the whole brain or everything that we know at that point from experiments that we actually put in simulation. But then you can also ask yourself, what is the point of that? You would really have to ask the really good questions in order to obtain information from that. I mean, you can do it. You can use it as kind of an experimental brain that you can't use right now. For example, human patients, you can't just, just take their brain and chop it up the way you want to. Of course, you use mostly non-invasive methods. And such a hugely, detailed computational model would permit such experiments because you could just change the parameters in the model.
Right.
I do personally like the approach of having a more simple model and understanding basic principles that are happening or that could happen in the brain from these simple models. That's the type of model I've been working on in the past. So, for example, one of the works I showed you that talked about the inhibitory plasticity in the synapses. Now, before I go into that, I have to explain what inhibition is in the brain.
Yeah, so, please do.
So you have a lot of different cell types in the brain. There are a lot of supportive cell types next to the actual neurons that transmit these electrical signals. But within the neuron population, we have neurons that can excite the next neuron that they're connecting to, and they're basically depolarizing the membrane of the postsynaptic neuron. And this is called an excitatory neuron. And the synapse of an excitatory neuron releases neurotransmitter that is then caught by the transmembrane molecules, transmembrane proteins of the receiving neuron in such a way that these molecules let ions enter the neuron that depolarize the potential over the membrane of that neuron.
And that creates
And
that sort
a
of
spike.
the electrical impulse or whatever you want.
So it excites the next neuron. It's quite intuitive. And inhibitory neurons, on the other hand, they have synapses that release a different type of neurotransmitter, different type of chemical, that leads to hyperpolarization of the postsynaptic neuron, usually through a chloride entry into the cell. And this... these two different cell types exist. There are also some that can kind of change their type or change their effect depending on circumstances or depending on development of the animal. But let's just say that for simplicity, we have these main two types, of which roughly, for example, in the cortex, I'd say roughly like 20% of those would be inhibitory and 8% would be excitatory neurons.
Okay. And the inhibitory ones, just to clarify, they're preventing the next neuron from firing?
So they could.
Okay.
They are actually, so it's basically, you have a balance between these two inputs. So you can have some neuron, some postsynaptic neuron, we'd say, like receiving a lot of different synapses. And it receives synapses from excitatory neurons, and it receives synapses from inputs from inhibitory neurons. And depending on how strong these synapses are, or what the activity of those presynaptic neurons is at any time, that neuron would be able to spike or not.
Okay.
So if the inhibition happens to be particularly strong at some point, then it doesn't matter how strong the excitation is. Maybe it just cannot make the neuron spike anymore because
cannot make it fire.
Exactly.
Yeah. Okay.
But if suddenly the inhibitory neurons are silenced, maybe through something happening upstream in the circuit, then suddenly the excitation can activate this neuron, and then it becomes active. So this kind of gating function is one of the proposed roles of inhibition in the brain.
Right.
So
it's a way of controlling which one fires, which one doesn't, and at which time.
Yeah, exactly.
And does it also control then, like, the strength of the firing?
Yes, exactly. So there's also
it can dampen the sort of firing of the neuron, or shut it off completely, or let it go through. Okay, that makes sense. It's like a control mechanism of how these things fire on and off. Yeah,
but it's there are. Of course, the picture is much more complex than this.
Of course,
it would
always.
be much too easy to just say that it would just be some kind of gating. There are many different types of interneurons in the brain that can be characterized depending on their morphology or their biomarkers, or like peptides that they have, and also the locations that they have in different parts of the brain. And in fact, this is one of the biggest fronts right now in neuroscience research, not just computational neuroscience in general. People trying to understand why do we have all these different subtypes of inhibitory neurons, and what do they do, and why are they even interconnected? So inhibiting inhibition. So there are already propositions coming that some of these neurons might be performing more like gating functions. Other are maybe more important for maintaining a healthy activity levels in the brain, by making sure that the excitatory neurons don't fire too much, so that you get an overactive brain, and so on. So it's really there's a huge potential for discovery here. Papers coming out from really big groups right now are looking into this, and one of the techniques that's being used in experiments is optogenetics. So the neurons express a type of ion channel that can be activated by light, shining light.
Right, I've heard of this.
Yeah. So and they're doing this now with specific inhibitory neuron subtypes. So they can say like, okay, we turn this cell type we turn off, this one we turn on, and we see what happens.
What happens? Yeah.
Yeah.
Okay. Wow.
So it's really quite advanced what's happening right now. But what I looked at before was I had a simple model in which I implemented plasticity in synapses belonging to both excitatory and inhibitory neurons.
Mm-hmm.
And I wanted to see what kind of plasticity rule would allow a balance between excitation and inhibition to occur in this neuron to prevent it from being overactive, for example.
Ah, okay. So what conditions would lead to, a sort of
Yes, exactly.
regulatory, like a nice balance? Because what would an overactive brain be? Like, would it
that
just
would,
be
for example, be epileptic.
Okay, there you go.
By overactive, of course, this is not an official term, but
Right.
Epilepsy is very serious disease. About 1% of the world population suffer in some way or another from epileptic seizures.
Oh, wow.
Yeah, it's quite
I didn't realize it was that high.
Yeah, it's really quite high. And epilepsy is characterized by seizures, and these are kind of waves of activity happening in the brain of a pathological nature. And the neurons, during a seizure, they can't function properly. They can't do what they would otherwise be doing. So if the entire brain, for example, is involved, then you're unconscious.
Right.
But also, you can also have overactive overactive neurons or seizures in subparts of the brain. So it doesn't have to be the whole brain that's involved. But one of the theories behind like what could potentially cause epileptic seizures or such pathological activity is a unbalancing of this excitation inhibition, so that maybe the inhibition is too weak or suddenly the excitation is too strong. The balance breaks down, and then these excitatory neurons start to become overactive. And that's the nature of this excitation. If you get one neuron that's super active and it starts to activate these other neurons that are also excitatory, then it can grow out of control very quickly unless the inhibition can stop that. Which maybe in some cases it can't, because these neurons could be perhaps the activity could be very weak, or the synapses have maybe decayed, or we don't really understand actually what is happening there. But it's one of the theories. It's much too easy to just assume that inhibition is actually just weak and therefore you get seizures. But it's one of the elements that is somehow involved in this change in its balance of excitation and inhibition.
Right.
So that's why it's important to understand how can you maintain this balance in the brain, what causes this to go wrong.
Right.
One of the other things that I should mention here, which is very important, is that I just spoke of activity and overactive neurons and so on. And we have, of course, also plasticity in synapses. Now, what kind of plasticity this is depends on the synapse type, but there is one type of plasticity which is called spike timing dependent plasticity. And it's a type of change in the synapse that occurs when the presynaptic and the postsynaptic neuron are active around the same time. Okay. And this could be they could be 10 milliseconds later or 50 milliseconds later, and there would be some change in the synapse. And there is actually quite a lot of evidence for one of these spike timing dependent plasticity rules that shows that the presynaptic neuron, if it's active and the postsynaptic neuron is active just a little bit later, then the synapse would increase in strength.
Okay, so it has like an amplifying effect.
Exactly.
Okay.
It rewards causality, and then it will, if it's an excitatory synapse, it will also increase this causality, because once the synapse is stronger, you will be much more likely again in a scenario where you have a presynaptic spike and then again a postsynaptic spike right after it.
Right. So down the line of the circuit, it could amplify.
It could amplify, and it could grow out of control.
Right.
So it's not just activity; it's also the plasticity that can play a big role into overactivity in the network of neurons. And for this reason, there have been people trying to model this actually using computational models to say like, okay, well, we know that this plasticity rule exists, but if we put it in, we just get out of control activity, which is like meaningless.
Right.
What do we do against that? How does the brain solve this problem? Because this is clearly not happening all the time. And one of the answers that we think is going on is that there is homeostatic effects happening in the brain, both at the synapse level and at the neuron level, that prevents these, at least in non-pathological cases, would prevent this kind of crazy activity. And one of them, for example, is called intrinsic plasticity. And in this case, a neuron has a certain spiking threshold. If you give this neuron, like if you depolarize this neuron enough, then it will spike. But if you stay just below this threshold, it will just not spike. And it's been shown that neurons can actually adapt their threshold over time. If they receive, for example, too much input, too many spikes, they can actually make their spiking threshold higher by changing the composition of ion channels and transmembrane proteins in their membrane.
So it'd be like kind of like overworking it. Like if you overwork it, it takes more to make it fire.
Exactly.
Right. Okay.
And conversely, also, if you decrease activity, then the neuron tries to adapt to that by decreasing the threshold, making it easier to spike. So there are mechanisms like that the brain has somehow implemented that work to prevent such pathological activity from happening. And another one happens, we think, at the synapse level, where the synapses themselves become stronger or weaker if the network activity is very high or very low for a long time. So there are all sorts of mechanisms that can act, and probably a lot more that we still don't know about, right?
Yeah.
So yeah, that's kind of like one of the big questions that I've been working on, that people in the lab here have been working on.
So this idea of like regulating it, keeping it in check, and so that's where you can have a picture of a sort of non-pathological brain, so one that's everything is being nicely balanced, inhibition, excitatory, and then using a model, you can say, okay, well, if we change this parameter, is it still in that homeostasis? Yeah. And if it isn't, then that gives you some indication that maybe that's what would lead to a pathological scenario. So did you find anything interesting there, or is it one of these like, well, we still looking?
Yeah. Well, in the specific case of epilepsy, like we're still working on that. But what's very important to know about these homeostatic mechanisms is the time scales. So a lot of experiments in the past decades that have looked at these homeostatic mechanisms that I just mentioned find that they work on very slow time scales. And if you make a model of it, then suddenly the slow time scale is not sufficient to suppress this activity. So you're like, okay, we need something faster.
Okay.
So that's the kind of thing that has been found like recently, that we need also some faster.
So would an example of a slow method of maintaining homeostasis, because like when we're talking about the brain, we can be talking about hundreds of milliseconds between neurons and stuff firing. So what is slow? What are we talking?
Slow, like hours, days.
Okay, so this is sort of like a long-term thing that's keeping your brain sort of in this normal range. So, but there's got to be. But when you put that in the model, it's not though. Yeah, it's clearly not the only thing that works.
No. There is some evidence also for faster homeostasis in the synapses on a scale of minutes, for example, and so. There is definitely probably a combination, like a whole zoo of these mechanisms acting.
Right. But with these, so like when we're talking about like a slow mechanism versus a fast mechanism, do we know what the mechanism itself is? Is it all about the levels of the different neurotransmitters in the brain? Or is it like you said, there's some of these structures that they have on the edges of the cells that they develop? Like, do we know that?
So that's a good question. So not everything is really known about how these operate, these mechanisms. So there is the mechanism of fast homeostasis on the synapses very likely has something to do with the maybe some limitations in resources of neurotransmitter, for example, or limitations in the receptors that receive these neurotransmitters in the postsynaptic part of the synapse. So some kind of redistributional limitation is happening there, but the problem is that it's different in different parts of the brain. So some experiments show that you have some kind of normalization in the outgoing synapses of a neuron, so that its outgoing synapses would pretty much sum to some value of a total strength of these synapses, and in others it would be the total incoming strength of a neuron is somehow kept constant over time.
Right.
So it's very hard to say like, okay, this is the exact mechanism happening there, but it seems to be quite connection dependent.
Okay.
Yeah, but what we've been looking at in relation to these homeostatic mechanisms was the possible role that they could play into shaping distributions of synaptic strengths that have been observed in the cortex and in the hippocampus. And one of the very consistent findings is that there are these long-tailed distributions with a peak at some small values of synaptic strength. So that if you look at some part of the cortex, for example, and you look at how big, for example, these synapses are, how strong, you will consistently find these long-tailed distributions. And this led to the question of like, so why do we keep finding these, and what's the function? And
so like when you say like the long-tailed distribution, it's more or less, it's like when you look at a certain section of the brain, there's usually the same sort of amount or proportion of synapses that are this big and this small.
Yeah.
We're talking about the sizes and the strengths of them.
Exactly.
Okay. Yeah.
So you see.
So this pattern is reoccurring, which makes you think that, well, there's got to be something to that.
Yeah, exactly. Well, it could be simply a result of some interactive variables that have some function, and the long-tail distribution is just a result of that. And we observe it doesn't mean that it in itself has some important function.
Right.
It could be like an artifact.
A byproduct of the other. Yeah, okay.
But of course, even a byproduct can be exploited over time through evolution, for example, to be helpful in the survival of an organism. So it's hard to pick apart these two elements.
But it's a nice finding to sort of give you the trail with which to follow, right?
Exactly. And so what's really interesting is that so in this long-tail distribution, in the tail, you have just a few synapses that are very strong. And in the rest of the distribution, you have many, many, many that are very weak.
Okay.
So you have kind of a rule of the few kind of structure there. And this is something that I've looked at in my work, and I've found that if you implement these plasticity rules in synapses, and you have some kind of distribution of some strong neurons, some weaker neurons, then you can actually obtain a structure in which these neurons that, for example, have higher firing rates, they also collect the resources for the strongest synapses over time due to this plasticity mechanism. So that's actually the paper that you mentioned also that you found. I call this the neurooligarchy, so that you have some neurons that they have some higher firing rates, or they receive more inputs than the other neurons. And if they are allowed to exploit this plasticity mechanism, for example, this spike damping-dependent plasticity mechanism that I spoke about, then they can get a lot of very strong output synapses by which they can influence the rest of the network. But the weaker firing neurons, their activity is much lower, so they can't really exploit as well these activity-dependent learning rules. So their synapses remain weak. And so you kind of create a structure of the strongest, most influential neurons in that circuit. And this leads to this kind of long-tail distribution of synapses, by which these strong synapses are properties of these highly firing neurons.
Right. So the sort of strong ones eventually recruit other strong ones sort of to them, and that's
Yeah,
you
yeah.
get this. They sort of dominate the circuit or the network.
Yeah, so you can make a step to like network science, for example, where you can where people look at, for example, the creation of like hubs in the network, like
Right.
very well connected parts of the network that can then also interconnect to create these so-called rich clubs of just it's the club of hubs that are interconnected and have a lot of influence on the rest of the network.
So this process, you've seen it in the modeling data that this is what, and you see it in the real brains that this so you've basically, your model has showed this is one way in which this clustering of strong neurons might develop in the brain.
Yeah, that's
Yeah.
yeah.
So then, if we have these networks of, or these clusters of strong neurons, and they're sort of, they're naturally occurring in the brain, they're somehow taking advantage of the homeostasis mechanisms or whatever, then obviously the question is, yeah, well, why has evolution allowed this to go on in the brain? Like, there's obviously I'm assuming there's some some functionality to having, a cluster of strong ones and then a bunch of other weak ones.
Yeah. Yeah, so that has actually been done by a former colleague of mine. He looked into this, and he found that you can, if you have such a structure of many weak synapses, a few strong synapses, and having this kind of structure in a neural circuit, then you can easily get a very precise propagation of sequences in the network, of sequential activity in the network. So you have just a few hub neurons that kind of dictate what's going on, and the rest kind of are just active enough to kind of support the propagation of the signal, but they won't really contribute to it themselves. So you get like reoccurring patterns happening. And another way you can also look at this is that if you have some strongly interconnected neurons that are mostly active at the same time, this is termed a neural assembly. And neural assembly is kind of a theoretical concept that we think might be very important for memory. It might actually be a memory or a part of brain activity that guides our cognition. For example, if you think of an apple, there's some neurons in your brain that are active right now.
Right.
That that are active every time you eat an apple or you think of an apple, and these neurons are very likely interconnected because they are active at the same time, and we know that through the plasticity rules, if they are active around the same time, then these connections will strengthen between each other.
Right.
So you can get what they call a neural assembly happening.
Right. So would this be the same as too, like when you're like learning a task, learning to write, or if you're learning a new hobby or something, music or something like this, you're gonna, the ones that are always firing when you're doing that activity will sort of create this, a hub of strong ones, and then the other ones, the weak ones around it, are there to just transmit the signal or whatever.
Yeah, it's not really like super solved yet, but that's kind of the theory behind it. Yeah, that you just have a memory represented by an assembly of neurons, and that neural memories would activate different sets of neurons. But we shouldn't think of it as in, oh, this neuron is part of this assembly, so it can't be used for any other memories anymore. Because if you look at experiments that look at how neurons respond to stimuli in for example, macaque experiments, that a lot of neurons respond to very different stimulations. So that you could show a picture of an apple, you could show a picture of a boat, and the neuron would still be active, and it makes you think like, okay, probably there exists many overlapping cellular assemblies that kind of borrow the same neurons over time, depending on the needs in the moment. And it's completely not clear how we should then think about neurons. We can't think anymore of the neurons as this neuron represents this one object or this one stimulation or this one concept that we have. Neurons are much more flexible than that.
So, so this one assembly or network of strong ones and weak ones is associated with one task, but it might also just be a part of another network that that you haven't even identified yet.
Yes.
So it's almost like if you zoom out, you might see that what you think is a main network, we'll say, is actually just a small part of another network.
Yeah,
so it just really depends on how you define the limits of your network, right? Oh, okay.
They could be like embedded networks, and
right.
in the brain you just don't only have different time scales, you also have different spatial scales. So you have neurons that are interconnected, but you have also groups of neurons that are interconnected between brain areas, and the whole frontal areas also connected to parietal areas. So you have these different spatial scales, and on all of these scales you have connections. So you have very long range connections, you have local connections, and you also have, there is also evidence that the neurons connect in certain patterns within the network, so that you have a lot of local connections with nearby neurons, and you have a few connections with neurons that are far away. This kind of network structure is called a small world network, and it's been looked at in the context of the cortex and the brain. And the advantage of having such a small world network is that you can propagate a signal through these neurons very quickly. So you can usually the path from one neuron to another neuron is quite short, but you only need a few long-range connections to do that. So you don't need so many neuronal resources. You don't need so much, you don't need so much pavement.
Right.
to which you would need if you were to connect all the neurons to all the neurons. Okay, the path length would be very short, but that would be impossible to still have a brain that you could carry in your head, like to say that.
Right.
Because it would require so many connections and so much insulation for all of these connections. So by only having a few long-range axons, kind of hypothesized like, okay, this kind of structure or similar network structure is probably implemented by the brain to make it easy to transmit signals quickly from one neuron to another.
It's just like
at the other side of the brain.
An efficiency thing.
Exactly, yeah.
Right. So, I mean, this is all, it's just amazing. And I guess this is why, the brain is so good at doing all the things that it does, all the time without us even thinking about it, like maintaining all the things going on in the body. We can think, like, it's just, we always think of it as such like an amazing computer. But so then these networks of things, these networks of neurons, and we're finding, how they're embedded now and all this stuff. It just makes me think so much. As a biologist, it's like the evolution of these things. Yeah. So do we have any idea, or is there any modeling? I mean, stop me if this is like totally out of your field, but of how, this might develop over time. Like if we look at simpler organisms or simpler models of it, is there sort of a set condition, I guess, in which these neurons sort of start to do this? Or is it sort of the Wild West and it can change all the time?
I find it very hard to answer this question. This is
no doubt.
I don't want to say anything that's like obviously wrong, but
it's okay. You're free to speculate here.
Well, obviously the structures that the brain evolves on, the brain evolves using structures that have been used by the previous organism. So you get kind of a structure built on another structure, built on another structure, built on another structure. It doesn't grow from scratch at every new organism.
Right.
So this will obviously create some limitations. Because we already have this part, we then have to make it work with this part. What I can say about it is that this will simply make it hard to understand the brain, in particular in the context of how we now use neural networks to create artificial intelligence, for example. We think like, okay, well, maybe we can create this kind of network that would be very efficient, and there are things that artificial neural networks, computers can do much faster than humans. So we can say like, okay, well, this is so optimal, and maybe the reason why the brain doesn't do that because the way it evolved. It's not just limitation in energy and size, weight, and resources and so on. I think it's also a limitation in what you already have, because evolution works that way.
Right.
And it makes it hard for us to understand, like, why does the brain do it this way? Why does it solve it this way?
Right.
Because we can construct something that's much more efficient at that task using a recurrent neural network. Might make
right.
Yeah.
I guess that's kind of the thing with any structure that you're looking at through the lens of evolution, it's like it depended on what the goals too were of that structure at that time.
Yeah.
You know, clearly the organisms that came before us weren't building computers or something, you know, so their network
Yeah.
at that point wasn't designed for that. Maybe it was simple stimuli as just light is here, light is not here,
Yeah.
smell something there, go that way, that kind of thing. So I guess it makes sense. So then it brings the question of, you know, understanding all this, modeling all this, and you briefly just mentioned it there, is what is it for AI? That's another thing that people are really interested in these days.
Yes.
So I don't know if you have thoughts on it or touch on it. Like, is this kind of, it seems like this would lead to, you know, if you understand these networks and how the brain is doing things, like you said, sometimes you can build things that are more efficient than the brain at a certain task. But I guess the goal would be to create something that's as good as the brain at doing all the things that the brain does.
Yeah. Yeah, that's something that, you know, we would definitely want to try and make. But I've lately I was thinking, like, you know, what is really intelligence? Because we define first intelligence based on, you know, the human brain. And like, okay, well, I know this person, he's very intelligent, or we know that the human brain can recognize pictures of cats very quickly.
Yeah.
And maybe we can call this intelligence. But then we're making more and more. I'm saying we, but just people in general.
Yeah.
Are creating more and more sophisticated algorithms that can and do more and more tasks that humans can also do, and others that humans are not so good at, making very fast calculations, for example,
Right.
involving many different numbers. Then you would have, I mean, it would be a problem then, because we would basically we would benchmark it with human brain, but then we are maybe creating something that's much more efficient or better than human brain at more and more aspects, which is only going to get better and better, I think, in the future. And then we would have something then, can we still call the human brain intelligent? That we have created something that's more intelligent, but how would we judge that?
Right.
You know?
Yeah, because like is the computer program that's really good at, you know, running the Google search, or like you say, doing numbers, is that intelligent compared to, you know, me being able to have a conversation with a number of different people at the same time? Or is that kind of just, is like there's different frames of intelligence? So if you make something that's super good at all these things, I mean, the natural inclination for me is just that anything that can do something faster than we can do it is more intelligent. But, you know, I see what you're saying. It depends on how you define it.
Yeah, I think this definition of intelligence, or how we view the word intelligence and its definition, is going to change in the coming years, based on, like, both what we understand about the brain, which is, you know, improving over time, but also what is, you know, being developed in AI right now. And I think it's only a matter of time that we will really start wondering, like, what is intelligence now, and how can we, like, you know, test human intelligence? How can we test machine intelligence? I don't know. This is more like a philosophical argument, but I've been thinking about that lately, and I'd like to think about, you know, what's next? What happens in the future?
Right.
Will the, you know, maybe some super intelligent AI with consciousness will then, you know, benchmark us against them, or vice versa, and so it's interesting to think of these things, like how the AI will actually evolve. Because the thing is that in my lab, so, you know, there are people working on neuroscience, but there's also people working on AI. So really specifically making kind of, you know, models that a robot could use, for example, to do a certain task, or trying to understand curiosity, exploration, so that, you know, it goes much further than just, you know, having a computer program solve some specific task. You would have some kind of independent AI entity, possibly, you know, put into a robot machine that could maybe move around, and then this would then explore environments by itself, and find out by itself what is important, what is really relevant in this environment. Maybe it will learn to open a door because there's a handle there. But how would you program AI to actually select these different parts? Would it
Right.
not? Would it not just look at anything randomly, unless you actually tell it beforehand, like, okay, well, there's a door
Right.
handle there, then, but that already defeats the purpose. We know
Yeah.
that human children usually, infants explore the environment by just touching things, and so there's people working on trying to understand this AI from this developmental perspective, like how can you actually move around, having this kind of AI that learns like a human, that you don't say like, okay, well, you have to press this button and then press this button next. No, discover by yourself. So
Right.
it's going much further than just solving a specific task.
But see, even then, it makes you think that like it's like a computational problem, right? Like say the robot exploring the room, you would still have to have an input in it that's like, rule 1, look around. Rule 2, touch things. Rule 3,
Yeah.
touch it in a different way, and like give it these. And I don't know, like, is that innate intelligence that humans have? Like that's how we do it. So is that the best way to do it? And how can we create something that's more intelligent than us if we're the ones designing it based on how we do it?
Yeah.
Right? Like, so is it, because it's not all just a matter of speed, right? Like speed of the circuitry and stuff.
No.
There's other things going on. So just, that's mine, I'm clearly just thinking about this right now and seeing where the train of thought goes. But how could you create, like, if we're always having the input and designing it and giving it its initial parameters, goals, whatever it is, how does it then become smarter than us? Is it just speed?
Yeah, it's a good question. I can't answer it. I can only like conjecture, like you are right now. But yeah, I think already there's been a lot of progress in AI and in, for example, deep learning. I'm not saying deep learning equals intelligence or anything like that, but once we get to a stage where so to speak, the computer can program the computer, then I think it's not impossible that we reach this stage of, that we go much further than our own intelligence.
Okay. So if we make it smart enough that it can, then tweak itself, do something to itself, it could maybe then see something, some pathway or some connection that we didn't, the missing piece that we didn't figure out, and then it would then become smarter than us.
Yeah, I'm thinking along those lines.
Yeah. Right.
Of course, researchers in robotics right now are mostly human, from what I know.
Could be some aliens in there that we don't know about. I don't know.
I think this will be the path along which they will go, like more autonomy for the systems, for the artificial intelligence system they're working on. This is not at all my field, so this is just what I've seen from or heard from my colleagues. But it's nice to see that people from like AI, robotics themed research are working together and working in the same place as people working on neuroscience, because we can borrow from each other, right? Like if you work in AI, you want to find some algorithm that can do something really well. Maybe you can, you're out of inspiration. Maybe you can look at how the brain does certain things, or maybe some properties of living neural circuits, and then implement it in your own algorithm. Maybe it will help, or maybe it will make it worse. You don't know that. You can try these things out. And on the other hand, if people in neuroscience want to try and understand the brain and looking at all these experiments and thinking like, doesn't make sense to me at all, and then maybe look at how artificial intelligence researchers solve certain problems, this can also inspire us. So I do still see it as two very separate fields, but there's a lot of potential for exchange and improvement here. Whenever I see a talk on artificial intelligence in some way, I'm always just amazed at like, wow, I never thought of that. And maybe we should, is this happening in the brain? Is it not happening in the brain? Will it happen in the brain maybe in the future? So this is a very important point that I think people should look at a lot, and like interdisciplinary exchanges between AI research or robotics and neuroscience are are very important for the future development of both these fields, I think.
Right. Yeah. I mean, I guess I always kind of thought that, AI research was, I had this idea of, for things like what you're doing, where you're modeling the brain, and sort of, if we had all the computational power, you could just model all those connections and know what they're doing all the time, and then you would basically just have, an electric brain. You could just, if we could just get all that information and sort it all out computationally, then you could just develop artificial intelligence, like it would live inside the computer or whatever, that it's all those, you're just simulating all of those connections. But is that really, I mean, say we had is that realistic? Would that then be an intelligent thing, or would it just be a replica of whatever brain that you mapped it on?
Yeah, it's again, it's also just philosophizing about this now, but
that's the fun part.
Well, the brain, as you know, is constantly changing. It's constantly, active in different parts. Different neurons are active sequentially. So you would have, if you have that program, you would have the brain at that moment,
right?
And you would have to have the same plasticity mechanisms, the same changes happening in those connections that would happen in the human brain, for example, the living brain, for it to be the same brain.
Right.
So if there's just a small change somewhere in one of the connections, maybe you just set like some of these synapses to not change, then already you'd have a different brain, and it would already be a different entity. Whether this would really be conscious, I really dare not mention anything about that. But I do think that people will try to do this, and they will succeed in a pretty near future, I think.
Because wouldn't, if you have this sort of hypothetical electrical, brain, whatever, living either, as a machine or in a computer, I mean, the biological environment must be was clearly playing a role in how the brain changes as well. So just by not being made of cells, or not being in a skull with air and different things, everything that we experience would already change it as well.
So there's a constant interplay between the environment and the brain. Your sensory modalities, picking up information from the environment. So without that, I don't think you could have real, conscious brain in an AI system. So all will depend on how you connect this to the environment. And
like if it lives in a computer, it's not.
But also like movement, visual system. I mean, the way that the eyes take up visual information from the environment, and how the visual cortex processes this information. This has all been adapted to the eye, to the retina, and if you change only a little bit the way these sensory organs work, I think this would at least you would probably need a very different structure or cortical structure to process this information well, and it will probably influence consciousness also, or awareness of this system.
So even just by like something simple, like something you wouldn't think of, just by it not having eyes or any of these things, it totally changes the nature of what the brain is and whether it's. Wow.
I think so. But
so,
again,
if you built it with say like a camera eye, not a biological retina eye, that would change the input signal coming in, the nature of that signal, and then all the changes that are downstream changes would be associated with that.
Yes, and you could different networks, different structures would be more optimal for processing such signals as opposed to the ordinary retina signal.
Right.
But again, this is really
far out there.
This is really on the not really clear what's going to happen there. Even just from the simple brain as we have it now, so much is still not understood.
Right.
Before we even try to move it to upload it somewhere.
Right.
On a couple of hard disks. We still have so much to understand still, and a lot of stuff happening also on the the cellular level. There's a lot of stuff concerning the maintenance of the neurons membrane, the synapses. So as I mentioned before, the lifetimes of synaptic molecules. We really don't know the details so much of everything that's happening there at any moment. So would we need to incorporate this, or we should definitely have at least some simplified version of it then in this artificial brain,
Right.
if we were to have such a thing. But then to what extent is it important to have everything there, and can you really simplify it? I think it would be hugely complex.
Yeah.
I'm not saying it's impossible. It will happen at some point in the future, but right now we're not ready to just code a brain just as like a real human brain in all of its complexity. There will be definitely a lot of loss of information if we do that now.
Right.
So yeah.
But you are saying that it will happen one day.
Yeah.
Okay.
I think so.
Okay.
I think it will happen one day. Yeah.
I mean, I'm not going to hold you accountable to that prediction.
Yeah,
but
I might be long gone by then.
But it brings up one other interesting thing for me in my mind is, okay, so we don't know, we can't replicate it perfectly. We can't, we're not there yet. But in terms of, we talked about epilepsy, but even just things like brain injuries or other sort of what we would say pathological things in the brain, this sort of information, modeling this, or being able to sort of understand these networks and everything that we've talked about would be a way to fix, say brain injury or,
absolutely
something like this.
Yeah. So, well, one of the things that we've been also working on is epilepsy in the brain, and I mentioned it before briefly. So, the epilepsy is currently treated in different ways. Some patients receive anti-epileptic drugs, and these work to suppress the seizures in some cases. But there are also types of epilepsy which are drug resistant. So even if you take a whole mix of drugs, you will still get seizures, and there is a lot of demand, to understand more how these types of epilepsy, how they can actually take place, how they affect the patients, and how we could potentially treat them in a different way. So a lot of these drug-resistant epilepsy cases are simply people who suffer a lot from seizures because the drugs don't work in that case, or the affected tissue is resected, so they just remove part of the brain.
Pretty invasive.
It's pretty invasive.
Technique. You have brain surgery we're talking about.
Exactly.
Yeah.
This is very serious. So. In particular, what's called medial temporal lobe epilepsy. It's kind of happening in or near the hippocampus. These type of epilepsies are often very difficult to treat with drugs. So we've been trying to create models actually to understand, what is happening in these types of epilepsy. By again creating a simplified model. It's only like very still in the early stages,
right.
But to understand. How could you have this process of epilepsy taking place on the circuit level? Like which neuron is active when? How does the seizure propagate? Which neurons are at the origin of the seizure? These are all important questions to find out if we want to find a way to really target this disease. So it's very hard to say something inclusive at this stage. But for example, there has been a previous work which we also try to adapt ourselves that looked at exactly these strongly interconnected networks, these hubs or interconnected hubs in a neural network that could potentially have an important role in the emergence or the creation or the propagation of seizures. So that if, for example, just a random neuron would suddenly become a bit overactive due to some changes in the membrane composition of the membrane proteins, for example, or it would become just sick. Let's just say it
Sure,
becomes sick and it becomes a bit too overactive. But nothing much happens to the rest of the network because this neuron has only a few weak synapses and it wasn't even that active before, so it's only a small increase in activity. Then that would not affect the network so much. But if you have a neuron that's already quite present in a network in sense of activity and the strength of its outgoing connections and its connectivity to other strongly active neurons, that could easily lead to overexcitation in the network. So there's some kind of thinking behind this, like probably these kind of interconnected hubs or some specific neurons might be actually the causes for these seizures, but we really don't know yet. We were trying to model this by understanding this. We're also looking at the role of some types of homeostasis. That homeostasis, as I mentioned earlier, can help to suppress activity, but it can also work in a pathological way, so that the mechanism is in place, but if maybe some neurons in the network die, that you have some remaining connections that suddenly become too strong, for example. That homeostasis would do this because it's just the mechanism perhaps is still in place by which this happens, but it would lead to a pathological effect. So there are some theories we're trying to explore here that homeostasis is not always doing good. It could also maybe play a role in epilepsy.
Right.
So that's kind of one thing also that we're looking at right now.
Yeah. So it'd be like you could also just maybe like if you had a better idea of how it propagated or whatever through or I'm thinking of one of those situations like there's these old games when you're a kid or whatever where it's like if you pull one stick out of the whole pile of sticks, all the marbles fall out. But it's like you can. The idea is that you keep plucking at one thing until the whole thing falls down, but you don't know. Like you're kind of.
I have no idea what game you're talking about, but I see what you're trying to say.
Yeah. But if you knew, don't touch this one, or we know that this one is the cause of it, then you could sort of, turn it off or attempt to do something like this.
Yeah.
To reset the network back into the.
Yeah, potentially, maybe, in the future, these more specific subgroups of neurons, or at least, smaller areas could be targeted, maybe even with gene therapy. So that, we don't have to resect a whole part of the brain, but that we can keep it and just make sure that the activity of some specific neurons is changed in such a way that you reduce the seizures. So you don't suppress the symptoms of epilepsy by just administering drugs, but you try to get at the root cause. And another thing that's also very important to know is that in epilepsy, in development of epilepsy, you often have a case where there is some trauma happening to the patient, and then after some delay, you would suddenly see seizures. But this delay could be weeks, it could be months, and it could even be years. And only after this delay, the seizures appear in the patient. So we want to understand what is happening in this delay period, and can we potentially stop epilepsy from developing during this delay. If we know what's happening, then we could maybe stop it even before it becomes like really detrimental to the patient's life by having seizures. So that's a really hot area right now in epilepsy research. People using both animal models and also reports from patients and recordings from patients. So some epilepsy patients have actually some electrodes, like invasive electrodes, for trying to find out the locus of epilepsy, like where the seizures are coming from, what is the affected area. So recordings from these patients are also used for neuroscience research sometimes. But this is still completely not known. We really have no idea yet what is happening in this latent period, if there even is a latent period, because maybe there are already so many changes happening that you can't stop them, but simply the seizures themselves only appear later.
Right.
So if there's any potential for treating the epilepsy before it actually becomes epilepsy, that would be really, really helpful, I think.
You would have to know then too the event that would cause it, right? So, and that's probably can be a number of different things.
Yeah.
So it adds a whole nother layer
Yes.
of.
Yeah, I thought of that as well. Yeah. So, well, you can have cases where, for example, patients have had a serious accident, and then
Right, like
if,
a head trauma.
yeah,
or
head
something.
trauma, and if the head trauma is sufficiently serious, then you. You know, the medical staff will then inform the patients, like, you know, you may be at risk of developing epilepsy, but maybe not.
Right.
It's not in every case. So this
Right.
is, again, something that has to be taken into account. But indeed, there are many other causes that could lead to epilepsy. So there's also like a brain inflammation, certain diseases, meningitis that could
Right.
lead to epilepsy. There's certain tumors. And the problem, though, in general, in the whole field of epilepsy research, is that if you see it like the patients that they have epilepsy, often the seizures, all the patients have seizures, but the way that they develop these seizures over time is probably very different, because all these causes are different.
Right.
And then we all call it epilepsy together, but then, you know, this is actually not the same epilepsy.
Right.
We can't,
subtypes of it.
Yeah, the subtypes of it, and this is very hard to find out, because the seizures themselves look very similar in all the patients.
Right.
Okay, so the periodicity is different, though. For example, some patients will have seizures maybe every day, and other people will have seizures maybe once every week or every few years even. So it can be a very long cycle in some cases. But the thing is that you can basically picture it. This is very well used analogy. Picture it like a river, and you have basically a lot of small rivers, and they all flow into this big river. And the small rivers, they all come from different places on the mountain, but at the bottom is a big river, and this big river is like the seizures.
Okay.
So the seizures, okay, we see them, like, you know, many patients, we see the same seizures, or many animals, for example, and then you don't really know what is the exact mechanism that causes this seizure to happen exactly at this time, under such circumstances, maybe involving these neurons and not involving these other cells. That is completely still open. And of course, you know, that in human patients you have non-invasive methods that are often used, so you can only find out, you know, something on the surface of the scalp. Yeah, there's a lot of things that make it very difficult. But this is like one of the hardest things about epilepsy, that we call them all epilepsy, in fact, and we know that there are many subtypes of epilepsy. But even within these subtypes, can you really say, you know, trauma, you know, in this patient and trauma, you know, in this other patient? Maybe they were hit on the head both at approximately the same area, but it's not exactly the same area.
Right.
You will never get that. And then it's very hard to say, like, you know, what exactly causes this epilepsy and how should you treat this patient. So based on, you know, previous experience of the medical, you know, experts, the clinical experts, they will, you know, you use their own experience, but it's very hard also to really, you know, treat this kind of thing. So there's a lot of, there's so much we still don't know about epilepsy. It's crazy. When I started working on it, I thought, like, okay, yeah, epilepsy, wow, there's a lot I have to learn about it. And already very quickly I hit this limit of, like, what, you don't know that, you know? oh, gee.
We don't even have that answer yet?
Yeah, exactly. I was quite shocked. So, yeah, I think it will be very important to combine both research from human patients, from clinical experts, with, you know, modeling, like things that we are doing, and also animal experiments that all come together with this. Then we can combine these things and really understand, like, what is epilepsy in all these different cases, and then hopefully treat it better than it's treated now. Yeah. So, yeah.
It's one of those things, I think, again, like when we started the conversation talking about how there's all these different aspects of neuroscience. I mean, I think that's a good point that you make, where it's like we kind of all have to work together. You can't look at it in isolation because there's so many levels of things that are going on. So that's really interesting. We've been going for just over an hour, but if you have a little bit more time, I'd like to ask you one more thing.
Okay then.
And this will definitely go more into maybe more of the philosophical stuff again. But as we're talking about all this stuff about, you know, understanding what these networks are and like how they're firing and what the signals that are going through, it just seems like there's so much noise potentially in the data, or so much data trying to decipher that. My idea, my like crazy whatever idea here that I'm having is, again, you can, with whether it's EEG or something, you can could you one day decipher based on, the firings in the brain, what that person is thinking? So maybe like mind reading is kind of a thing, but I'm kind of thinking more, I'll give you the reason why I'm thinking this: dreams. I think they're, it's super fascinating. We don't really know why people have them. And sometimes I have these really vivid dreams, and I want to wake up and I want to be like, oh, if I could only like tell somebody what that was, or describe it better what it was.
Yeah.
But if you could, get all the signals from my brain as I was dreaming and actually turn that into the picture that I was seeing, or the memory, I guess, as well. Like, is that even possible? Again, like if we had like all the computational power or whatever.
I think I remember seeing a paper somewhere that actually did something similar to that.
I've seen this, but then I saw the pictures and I was like, I wasn't totally convinced.
It was very still. I mean, it's a great effort.
Yeah.
But it's not as sophisticated as what you're suggesting now. So, there's obviously been work where people, told participants, like, think of a fish, for example, and then record your brain activity. Then sometime later, it's like, okay, think of something, and then look at your brain activity again. Say like, okay, well, based on decoding the information that we just got from your EEG or activity, for example, or your MRI. I don't remember exactly what it was, what they recorded, but based on your brain activity, you can say like, okay, you were thinking of a fish,
right.
But that they only had a specific set of things you could choose from, right? You could think of these one out of 5 objects or animals that we tell you, and then you can decode it, but it's not like we can tell exactly what you're thinking. But in a more general way, the neural code is still not understood completely. Like the neurons, they're firing, and if they're firing together, you can kind of observe these brain waves, these oscillations in the EEG. But we still don't really know what these signals exactly represent.
Right.
So you could do this kind of, just broad, like this, recording the whole brain and then putting it through like some sport vector machine or some other algorithm and trying to predict like which out of so many categories you actually were thinking of.
Right.
But then you need a lot of data from that one specific person.
And then everyone's going to be different.
Everyone's going to be different. You have that problem as well. But what I personally find interesting is really just understanding this neural code. Like, what is a spike? Like, what does one spike mean?
Right.
In many cases, we still don't know what this really means. Okay, so it's a signal propagated from one neuron to the next neuron, and as we spoke about before, like one neuron may be part of some assembly of neurons at one moment, part of another assembly at other moments. So it's just so complex. We really want to find this out, and what's really even more comes on top of that is then the plasticity and the activity interacting, because they are both interacting constantly. You have this feedback loop of plasticity dictating what kind of activity will come next, and then the activity influences the plasticity.
Right.
So it's also constantly changing.
Yeah. Right.
Yeah.
I wish.
Tell me what you dreamt last night. Maybe I can look at your brain activity later.
It just seemed like with the modeling stuff it's just such a really cool approach to, because you're really you're building something that's like what you're seeing, and then you're able to sort of interpret that. I don't know. It's a really interesting approach, I guess, rather than just like getting all the signal and then trying to make sense of it. It's like, well, no, let's build it, and then that will let us know what makes sense or not. Yeah. Very interesting. Yeah. Well, I mean, that was really great. Super. So unless there's something else that you really want to touch on or anything, I think we can wrap it up.
I just wanted to mention that I developed this online course together with my supervisor, Professor Joachim Trisch. And so maybe this course will be put online very shortly. So maybe you could provide the link.
Yeah, definitely.
On
We
your
can talk about that
podcast.
after,
Yeah.
and I'll put the link out. I'll tweet it out. If you have any,
That'd be
Twitter
really
or
neat.
something
Yeah.
you want to tell people about, go for it. Yeah.
Yeah, it's just an online course to learn how to program your own neuron. In the style of these neural circuits that I mentioned before. So you would learn to program your own neuron, and you could make it your own network by using these neurons and put plasticity rules in it. And I explain these plasticity rules also as well in this course. And the course is open access, so you don't have to pay anything. So
wow, great!
I hope it
So
will help some.
It's a whole interactive program. Build your own neuron, and it's just available online.
It will be available online when you put the link up, and it will be open access on the website. I'll tell you about the website later. So,
okay, perfect.
But so if you're interested in making models yourself, models of neurons, models of plasticity in the brain, then I really recommend you check out this course.
Great, that's awesome. That's such a great, to just do that. Did you just do that in your spare time? I'm just gonna whip up this module and let people see it.
Not spare time, no. Time, yes.
Yeah. Excellent. Okay, that's really great. We'll definitely put that out there and let people know about that. Thanks again. This is a great conversation. Maybe we can do it again sometime when I have more philosophical questions about the brain that I want to tackle.
There's so much we can think and talk about still. But thank you very much, Brad. It's been really nice. It's been a pleasure to discuss these issues with you and these fronts of the neuroscience research. Mm-hmm. And I hope that we will keep on asking questions about this, because there's still so much to be discovered.
Definitely. And like I said, people love learning about their brains. People love hearing these crazy things and about AI and all the rest of it. So again, thank you so much. And
thank you.
Yeah, we'll leave her there. All right. Wow, there you have it. What a whirlwind tour through the brain. We'll have to do that again sometime. I want to thank Florence again for joining me. That was such a super fun conversation. The course that she mentioned online at the end there, as far as we know, it's not up yet. I will keep you posted on the social media channels and in coming episodes as to when that does get posted. So if you are interested in that, stay in touch with me on Twitter, either from the show account at 2brad4u or my personal account at bvanperdon, to get information on that. And if you're a regular listener to the show, then we will update that continuously, so don't worry about that, missing it then. I guess other than that, if there's topics that people want to cover, get in touch. Again, either Twitter or Instagram even. I'm on Instagram at bvanperdon, at 2brad4u. It's all the same. Because then I can seek out people to have those discussions. So if you are listening, if you do want some specific topic, let me know. Many thanks to the Freak Motif. They just played a bunch of shows in southern Alberta and BC, I believe, back home in Canada, for their album release, Hot Plate. You can listen to it on Spotify, or go to their website. We have links to it all on our website at 2brad4u.wordpress.com. Check it out, check them out. Great band. Thank you to them as always for letting us use the music and being fans of the show. Other than that, I think that's everything. You can check out the Tragically Hip podcast, cast of the Unplugged Gems. I'm part of that with a number of great guys, breaking down hip music. If you're in Canada and you're a hip fan, check that one out. You can follow it on Twitter at Cast Gems. You can get it on iTunes, Stitcher, all that good stuff. Okay, I think that's it. I think that's enough of me promoting all of my things. Thank you again to Dr. Florence Kleberg. It's a real treat. And we'll be back next time with more episodes of conversations, or Brad and I having conversations. Until then.